Hi, everybody. Today we're going to talk about DNA replication, or the copying of DNA. Now, you'll remember that when cell division, mitosis or meiosis happens, you need to make identical copies of the DNA so that each daughter cell can get an exact copy, right? Remember that every single cell in your body, from the top of your head to the tip of your toes, except the sperm and egg, have exact copies of DNA. Well, every time that the cell goes to divide, you have to copy the DNA during interphase so that there's a copy to go to one cell and a copy to go to the other. And now we're going to talk about the copying. Now, remember I said that in order to understand the functioning of DNA, you needed to understand its structure. Well, this is where this is going to first be important. DNA replication, the copying of DNA, happens at the beginning of cell division. And it happens in the beginning of both mitosis and meiosis. You need the sister chromatids, the identical copies, connected to each other. Remember, we drew them as an X, right? And they were connected at a centromere in the middle, right? And they're identical to each other. Those copies have to be made, whether it's mitosis that's dividing them or meiosis that's dividing them. And so at the beginning of both mitosis and meiosis, you have DNA replication. So I'm going to explain how that happens today. Now, if you remember, DNA is two strands, right? You've got a strand of nucleotides on one side and a strand of nucleotides on the other. They're like a ladder attached in the middle with the nitrogenous bases, and the whole thing is twisted up. And remember, that's all held together by loose hydrogen bonds. So it can zip up and unzip, and zip up and unzip. Okay, loose hydrogen bonds holding it together. That zipping is really, really important for replication. It's here that the DNA will unzip, and you'll have one strand of nucleotides on one side and the complementary pairs in a strand on the other. So if there was an A up here, there's a T where it used to attach, right? So they're complementary to each other. So it unzips at the weak hydrogen bonds. And now you have these two separate nucleotides. So we've got our two separate nucleotides. Let's say this was A and this was T. They're separate. Now, floating around in your nucleus are bunches of nucleotides. If you have the right enzymes to attach them in, if you have an A right here and a T right here, and there's a T floating around in the nucleus, it can float in and attach to the A. And if you have a T, it can attach over here. So you can get an A and a T and a T and an A. And so that will start to happen and match. So if you look here, you can see in number 1, we've got the original strand of DNA. First thing that happens is it unzips. That's what's happened in step 2. You now have an old nucleotide strand here and an old nucleotide strand here. That is simply what I was showing you here. The DNA, the double strand, unzips at the weak hydrogen bond. So now you have two separate old strands of DNA. You also have floating around in the cell, these little blue things down here are new nucleotides that have been floating around in the nucleus. Basically, you've eaten food and building blocks you've digested and you've brought into your bloodstream and brought to your cells, and now you have these things floating around. So if this right here at the top of the left-hand strand was a T, and if this is an A, it can match. Well, if this was a T, that means over here was an A. That means if this is a T, it can match. So if we look over here by step 3, this old strand that was originally part of the original DNA over here now has new complementary pairs attached to it. And if this strand right here, which is here, is over here now, its order of bases is going to be complementary to this. And how will the two new strands of DNA compare to each other? They will be identical. The ones in red over here are the old ones. They started here. They're just now, this is the half of the original old one. This is the other half. And we have two complementary base pairs in the middle.